Okay, hello everybody. We're going to wait just a few minutes and allow folks to start rolling in. Um, we can watch the count go up as you all are able to get into the webinar. And we're really looking forward to tonight's webinar. This is a treat for us to have Chris, and I am anxiously awaiting what he's going to be sharing with us.
Thanks, Connie. I'm
You-
excited to be here. I can't wait to speak to this group.
You're welcome. Are you in Salt Lake right now?
I am in Salt Lake. I'm in an office that I spend a lot of time in, surrounded by whiteboard walls, where we try to ideate all kinds of interesting science.
And how's the weather? Ski weather?
You know, it snowed a lot this week, but I have only a little tiny window I can see. It looks like it's fine right now, not even raining.
Excellent. Excellent. Well, I think we are at almost critical mass here
Great.
for starting. So I'm just going to start with some housekeeping items. This is a webinar in which Chris will do a presentation, and then after that, we are going to have a period of time where folks can ask questions. What we would like for you to do is look for the button, the box on your screen that says Q&A, and click on that and type your question into there. If you type it into the chat, very good chance that I will not see it. So please use the Q&A when you're asking the questions. The other thing here is that this webinar is intended to be for our patients. That's why the Alliance to Cure is hosting it rather than Recursion. It is for the patients. I understand that we have a lot of different kinds of people attending right now. When we get to Q&A, I get to curate the questions. It's my job. And I'm going to make sure that the patient-oriented questions are asked first. If we have time still after that, we can try and throw a few others in, but the patient-oriented questions are the most important. This is their opportunity. And then finally, we have our support group this evening after the webinar. And Darla, I believe, is here, and she's going to be throwing the link to the support group into the chat at the probably now, and then again as the webinar goes on, and definitely toward the end. All right, now it is my pleasure to introduce Chris Gibson, and I have a whole big write-up of who he is here that I get to
But
read.
you're going to shorten it down for everybody.
Yeah, sure, I am.
Please.
Chris is the co-founder and CEO of Recursion. And just for a little bit of background, he developed the technology and approach that seeded Recursion as part of his MD Ph.D. work in the lab of co-founder Dr. Dean Lee while he was at the University of Utah, so as a graduate student. After completing his Ph.D., Chris left medical school to build Recursion into the rapidly growing company it is today. I'm sure he'll tell you more about the company's mission and platform as part of his presentation, but he has Recursion's headquartered in Salt Lake City, has offices in Toronto, Montreal, New York, London, the Oxford, UK area, and the San Francisco Bay Area. He serves on the board of BioHive, which is a public-private partnership driving expansion of Utah's life science ecosystem. He's also active as an advisor and mentor, both formally and informally, to many young biotech founders. He truly is a mentor at this moment. As young as he is, he's a mentor. Finally, he enjoys cycling on both the roads and the trails that cut through Utah's great wilderness, as well as spending time with his family. Chris, I'm going to let you take it away from here.
Oh, thanks, Connie. Thank you so much. It's been amazing. I think, Connie, we met for the first time back in 2011, maybe in Paris, something like that.
Yep.
It's been an incredible relationship, and the Alliance to Cure has just been such a fantastic partner. And so I feel really privileged to be here, getting to speak to the patients this evening. It's a real honor for me to be able to do that. I do want to share that I will talk a bit about the company and the mission, but I'm going to really focus on sharing exactly what we shared when Dr. Burkhart presented this data at the International Stroke Conference. One of the opportunities and challenges of being a public company is we have to make sure everybody has the same information at the same time. So you'll hear from me instead of Dr. Burkhart, but I'll be able to share really the same results that were presented there a few weeks ago. So let me dive in. As a public company, I need to just share that I may say things about the future. And it's important for you all to know that I don't know what the future looks like. And so these are all just my perspectives on what the future may look like. So please take them with a grain of salt. So really delighted to share this story of a phase 2 randomized placebo-controlled trial evaluating Rec 994 in patients with symptomatic cerebral cavernous malformation, as presented by Dr. Jan Karl Burkhardt at the ISC meeting a few weeks ago. And I'm going to take a little bit more time since we all have time together to go through it and talk through some of the data in a bit more depth. I think Dr. Burkhardt had 12 minutes to get through all of this. Since we have so many patients here on this webinar, I should be hearing from you about the way this disease affects you. But for those who aren't patients or are new to this journey, I think maybe I'll give just a very, very high level overview. This is a really, really important disease that I think is becoming more well understood thanks to the work of Alliance to Cure and many others. But really is a pretty widely prevalent rare disease with 360,000 symptomatic patients in the US and Europe, and 90,000 patients in the US and Europe who actually have lesions in their brainstem, which is one of the places where these lesions can be most problematic. So this is among the rare diseases. This is a relatively common disease and one that I think we want to keep getting the word out about. As many of you know, this disease is characterized by the formation of these vascular malformations, and you can see on this MRI these vascular malformations look like these little circled holes here. These are small capillaries, the smallest blood vessels in our body in the central nervous system, so the brain and the spinal cord. They turn into these berry-like vascular malformations. And over time, those lesions can leak and they can bleed. And over time, that can lead to lots of different debilitating symptoms. So seizures, if some of these lesions are in the cortex, or maybe focal neurologic deficits, if some of these lesions are in other parts of the brain. And over time, in patients that have a single lesion, that lesion can grow. And typically those patients are what's called sporadic. So they just get one lesion. And then there are patients who have a familial form of the disease where they have someone in their family who passed on a gene that increased the prevalence of them, or the probability of them getting not just one lesion, but in some cases hundreds or thousands of lesions. And those patients can go on to see lots of those lesions grow and worsen over time. And so really important disease for the world to be focused on. And up until now, there's been no company that has done an institutionally sponsored clinical trial trying to take a medicine forward for the treatment of this disease. There's been lots of great work in the academic world, but in order to get a medicine all the way to patients, often it takes a company to come do that. And we're trying to build a pathway. I will say before I dive into some of the data about the clinical trial, these clinical trials are early and they are hard, and we are the very first company to do a clinical trial with the FDA in this space. So a lot of what we're doing is blazing the trail. And what's great about that is whether or not our medicine ends up working, everything that we're learning along the way and everything that the FDA is learning is going to be helpful to any companies or any institutions that come behind us. And so I think that is, one of the most fulfilling parts of this is success or failure. We're going to be helping to move the field forward. I also want to just share before I dive into some of the data on this clinical trial that it's very early and we're seeing encouraging signs on certain measures that I will talk through, but by no means are the results that I'm going to share definitive. There's tons more work to do. We have Beth Bruckheimer and Janine Fogley from our team on. Janine leads a lot of our efforts with patients, and Beth is the lead of this particular program. And I might call on them to help me during the Q&A period if needed. But I want to acknowledge the incredible work, not only of Beth and Janine, but of the almost 800 people we have working at Recursion now. We've got medicines for eight different clinical programs ongoing, and dozens of programs coming behind that. And we're working really, really hard to change the pace, the scale, the probability of success of medicines getting to patients. And this is one of the programs that we're really, really proud of, and one of the programs that's most advanced. So now I'm going to take a step back and I'm going to go all the way back as Connie aged me back to graduate school to the work I did in the lab of Dean Lee at the University of Utah. And this was back, this paper came out in 2015, but a lot of this work was done sort of 2011, 2012. And it was, relatively novel at the time. We built cellular models of this disease. So we took a thing called siRNA, which is a little tiny nucleotide that you can put into human cells, and it will go essentially soak up the message that encodes the proteins that are encoded by the different genes in CCM. And in this particular case, we soaked up the message for the proteins that are involved in CCM, and specifically CCM2. And what we can see when we look under a microscope is if we use a control siRNA, this is what a microscopy pictures, a picture of these endothelial cells look like, and they kind of look like cobblestones. And you can see there's a nucleus in the middle, and then there's these cell borders. And these look to somebody who looks at these a lot like what we'll call quiescent or unactivated, or we'll just say kind of happy endothelial cells. When we knocked down the CCM2 gene, so we reduced the amount of the CCM2 protein in these cells, where they start to look what we'll call activated. And so you see that there's all of these green things. These are called actin stress fibers. There's changes in the shape and the size. And it's really easy to look at an image like this and say, oh yeah, let's measure the green stuff. And we had done that in Dean's lab. And we found medicines that reduced the green stuff in these cells. A great example of these is simvastatin. And ultimately, when we went to animal models and beyond, we didn't see that those molecules were translating. And so we tried something new where instead of having humans look at these images, we actually trained really, really basic machine learning classifiers, which you can think of as like very small versions of some of the AI tools that you hear about talk today. And these machine learning classifiers would look at thousands of examples of the disease cells, thousands of examples of the healthy cells, and they would build for themselves a mathematical model of what made these images look different. And we then put thousands of drugs onto these disease cells. We took pictures again, and we asked the algorithm, do any of these drugs make the cells look healthy again? And this is a picture of these same disease cells. So we've knocked down CCM2 here with REC-994, which is the drug we're talking about today. And what's fascinating about this is, to me, these images don't actually look, see all the green stress fibers everywhere? They don't really look like they're completely fixing these cells. But according to the computer vision algorithm, which is seeing lots of things that we're probably missing as humans, they might actually be. So a good example of this is you can see here, the cell-cell junctions look all red, whereas in the healthy cells, you see a lot more yellow. And you see over in these REC-994 cells, you see more of this yellow. Maybe that's something that the model was picking up. And ultimately, we looked at thousands of molecules. We found a few that looked really interesting, and REC-994 was one of those. We did not know what REC-994 was doing, nor did we look at a lot of medicines that were doing specific things because we had read all the papers and believed that this was the right mechanism. We looked at thousands of molecules that had hundreds, if not thousands, of different mechanisms or targets, and we just let the images guide us. And ultimately, what's really interesting about REC-994 is that it turns out this is a molecule that soaks up these things called reactive oxygen species, and in particular, this thing called superoxide. And so when we found REC-994, we were a little bit surprised. It turned out there had been a couple of papers about this from a laboratory in Italy, if I remember right, Francesco Retta's laboratory, but we really hadn't been working on this, and it wasn't a popular area of study in the field. But as we started exploring this early hit, we found if we took endothelial cells and we broke one of the genes associated with CCM, like CCM1 or CCM2, when we would measure the superoxide in these cells, it was really, really high. And we could actually trace this back up to changes in a transcription factor called FOXO1. And so this was interesting. We found a molecule that we knew soaked up extra superoxide, and that was a surprise. And when we looked at the cells, we saw that indeed, if you modeled CCM, the cells had a lot of extra superoxide. So that was an interesting and important connection. What was also interesting for us is that there is a lot of literature on the role superoxide and other reactive oxygen species play in the endothelium. And the endothelium are these cells that line our blood vessels where these lesions actually grow. And it turns out that too much, you need some of these reactive oxygen species, but if you have too much, your blood vessels can get leaky. and they can get inflamed and activated. And that is in some ways what we see with CCM. And so this was a really interesting hypothesis, and we pursued it further. And we pursued it further. I'll actually go on the right first here. We took mice where we modeled CCM in various ways, and we asked whether or not we could see something in these mice that we could test our drug against. And what you see here on the right is an assay where we took mice where we modeled CCM disease, and we took healthy mice and we measured how much leakiness they have in the blood vessels of their skin. And so we put a dye in their blood vessels and we see how much of that dye leaks out into their skin. And you can see that if you take just a control or Rec 994, there's like some amount of leak. All of our blood vessels leak a little bit. That's how nutrients and other things get in and out. If you look at a mouse where we had knocked out CCM2 in the endothelial cells, you can see that with a vehicle, so this just means a control, you can see that there's a lot of extra leakiness. So these mice had way more leakiness in their blood vessels. And when we gave them Rec 994, you can see that it rescued this leakiness back to a level that was similar to what we see in healthy, normal mice. And so this was really interesting to us. And so we did a bigger, harder experiment where we gave mice Rec 994 for 5 months. And then we used little tiny MRIs. So you've all seen the big human MRIs. We used little tiny mouse MRIs, and we looked at CCM mice that either had been given a placebo, essentially, like a mouse placebo, which we'll call vehicle, or they had been given Rec 994. And what you see here is that at 5 months, when we looked at these mice, you can see that the lesion number was reduced when you treated them with Rec 994. And if we look at the size of lesions, we see that we were reducing lesions, lots of these small lesions. In purple, you can see there's less of these lesions than you see in vehicle. There wasn't really a difference that we could see among the large lesions. Like many of these mice that we gave the drug to still had 1 or 2 of these large lesions, but we were reducing a lot of these smaller lesions that were growing. And so this was really promising, and this is what led us to get excited to continue the work to move this molecule into patients. And so it took us several years. We were a small company, originally funded by NIH grants and then bringing in investment dollars. We didn't even start working on this particular molecule for clinical development until 2016, because we had to assure the company was in a good spot and had funding and all of that. And we had to go do preclinical studies and do what's called an IND. So we did lots of tests to make sure that this molecule was something that would be safe. And we put together what's called an IND enabling package. And we went to the FDA and we said, we want to run a clinical trial. And then we ran a phase 1 clinical trial, which we've previously talked with many of you about, where we looked over a small number of days at whether or not healthy volunteers, so patients without or people without CCM, did they have any kind of health issues when we gave them this medicine at varying doses? Did they have headaches? Did they have back pain? Did they have nausea or vomiting? And what we found was that the molecule was pretty well treated. And so next we got to do what's called a phase 2 trial. And it's important for me to note here, we're the first company that's ever gone to the FDA to do a phase 2 trial. So there was not a clear regulatory framework for us to work in. There wasn't somebody at the FDA who could say, hey, another company just ran a trial here and we previously negotiated with them what the best things to measure are. You should replicate that. We actually got to work with our partners at the FDA, and they are fantastic partners, and with Connie and the Alliance to Cure and the team there, and many patients that were involved in giving Connie and that team feedback on designing a trial that would help us do 2 things. One, it would help us learn about Rec 994, but also, and importantly, it would help us learn as a community, as an industry, about all the different things we could measure in this disease in human patients, so that regardless of whether Recursion was successful with this clinical trial, there was a pathway for the next company to be able to pursue a medicine and be able to do it more efficiently. And the way we decided to do this was with what's called a randomized, double-blind, placebo-controlled phase 2 trial. And what that means is that the doctors who were working with these patients and the patients were blinded. They didn't know if they were getting high dose, low dose, or placebo. And we randomized patients so that no patient knew ahead of time what they were going to get. And we had 20 patients on placebo, 21 patients on the low dose, and 21 patients on the high dose of this particular of Rec 994. And we treated them for 12 months. And we did visits and did gather data from these patients for those 12 months. And at the end of 12 months, once we'd gathered all that data, patients were able to opt into an extension study, which we essentially say, if you've tolerated the medicine well to this point, or if you've been on placebo and you haven't had the medicine up until this point, now we're going to unblind you, we're going to give you medicine, and you're going to be able to continue on the medicine to generate more data for us, because it would be potentially unethical for us to take patients off the medicine if it was being well tolerated before we had some sense of whether or not it was working. And I will just share, this extension study is ongoing. So there are still patients from this trial who are on this medicine today. We did lots of work during the trial. So we looked at MRIs at various points. Or I should say, before patients came in, we confirmed that they had a lesion by MRI. We made sure that we knew something about their symptoms being related to one of those lesions. They had to have a modified Rankin score, which is an assessment that a neurologist or a neurosurgeon might give to a patient based on their functional limitations associated with this particular disease. And then as patients went through, we looked at how safe the drug was, how tolerable it was. Did it make people feel bad? Did they get kind of safety issues? And then we also looked at a whole bunch of secondary endpoints looking at potential efficacy. So, for example, did we see a change in neurologic disability with this modified Rankin score? Did people get better on that? Did we see a change in disease-associated symptoms or patient-reported outcomes? Did we see a change in MRI, size of lesions or number of lesions? Did we see changes in hemorrhagic events, et cetera? And so I'll go through some of the results that we found here now. Before I do, it's important to note that this was a small study. We didn't have the money or need to power this as a very first study to give us definitive answers across everything, because that would have meant more and more patients and more time. Instead, we took this small group of 62 patients and we asked some initial questions that can help guide potentially other questions we'll ask down the road. And what's important to note here is, just because we randomized patients, you can see that the different groups were relatively similar, not perfectly similar, but relatively similar. So for example, the average age of patients in the placebo group was 47 years. It was 46 years in the low dose group, and it was 48 years in the high dose group. And so you can see, for example, if we look at body mass index, it was 28.1 in placebo, 27.6 in low dose, and 27.1 in high dose. And it's important to make sure, so you don't lead yourself astray with statistics down the road, that you balance these reasonably well. So you wouldn't want all of your older patients in the placebo group and all of your younger patients in one of the treatment groups, for example. And I think we achieved that for a small study. The next thing we looked at was the most important initial finding, which was looking at safety and tolerability. And this, chart is a little bit confusing, but I'll walk you through it. Here we're looking at placebo, low dose, and high dose. Now remember, when a patient was in this study, neither the patient nor their physician knew whether they were taking the drug or they were taking a placebo. And so patients would have, let's say, a headache. And if they had a headache, regardless of what group they were in, and nobody knew what group they were in, we would measure it. And what you can see here is any treatment emergent adverse event. So this means they're given the treatment and something changes. They have dizziness or headache or back pain, or they have flatulence or nausea, vomiting, whatever it is, we would measure those. And what you can see is that the number of patients who reported something that didn't feel right with placebo, with the low dose and the high dose, was pretty similar, 17, 18, and 15 across these groups. And then we looked at some of these things that are a little bit more serious. So not just like, oh, my tummy hurts, but like maybe somebody has a little bit more serious of a symptom. And you can see here, 4 patients in the placebo group, 7 in the low dose, and 3 in the high dose. Again, pretty similar across these different groups. And then what we did is looked at what's called a treatment emergent adverse event related to study drug. And what this means is that a physician who's working with this patient says, I think that there's a high chance that the patient is actually having a symptom that is related to whatever it is they're taking, be it placebo or the drug. So if a patient scrapes their arm on the door going in and out of the room, probably that's not related to the drug. But if a patient gets dizziness 15 minutes after they take the drug, that might be really obviously related to the drug. And so you can see that there were 2 patients in the placebo, none in the low dose, and 5 in the high dose that had something that we considered, or the clinicians considered, was related to study drug. What's great here is you see none of these things are greater than grade 3. So no patients came off the study because they had some side effect that was related to the drug. This is, as far as clinical trials go, it's very normal for us to see, every day, some of us wake up with a headache or dizziness or back pain or constipation. And so you always see some of these things in the placebo group. And what we really concluded here was that there wasn't a really meaningful change in the way patients were feeling from placebo to low dose to high dose. And that's great. Over a 12-month treatment, which is a long time for a clinical study, we felt like, at least in this set of 62 patients, this was a pretty safe and well-tolerated drug. And that's really good, because if it wasn't, that would present a lot of problems for us going forward. Next, we wanted to look at MRI. And the reason we wanted to look at MRI in these patients over the 12 months is that it's really objective. People can feel different ways. Physicians can come in, grumpy one day and happy another, and there can be a lot of variability in their assessments. But MRI is very objective. We use this machine to take a picture of people's brains, and then we can use machines to help us measure the size of these lesions. And so I won't go through the details of how we did this, except to say that we gave all of the images to a radiologist who was also blinded. So the radiologist did not have any knowledge of what drug the patient was on. And it was the same radiologist for all of the images. And so if they had some bias to measure a little bit more in certain areas, then that bias should be spread across all of the different patients. And this was encouraging. It's not definitive, but encouraging early data here that we see. And so let me walk you through this. This is called a waterfall chart. And what we see here, let's look at just placebo first. Each of these bars is a different patient. And 0 would mean from the start of the trial, before a patient took the medicine, until 12 months after they finished taking the medicine, their lesion didn't change at all. And so what you see here is there was a patient who we found out later was on placebo, whose lesion got about 20% smaller over that 12 months. And then there was a second, a third, a fourth, and a fifth patient whose lesions got a tiny bit smaller. But then the majority of the patients here actually had lesions that increased a little bit in size. And one patient had a lesion that got, roughly like 12 or 15% larger. Let's go look at the high dose here for a minute, because I think that's where things are most pronounced. What we see in the high dose is actually there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 patients out of the 21. So nearly half of the patients who saw their lesion get smaller over the course of 12 months. And you can compare that here with 1, 2, 3, 4, 5. So roughly 25% of the patients in the placebo group. So basically twice as many patients in the high dose treatment group saw their lesion decrease in size. Now, what was a little confounding is that most of these other patients, they kind of look like placebo, like there's a small increase, but there was one patient that had this really big, like 45% increase in their lesion. Even though that is true, when we look at the overall, looking at all the data from these patients, the placebo group saw an average mean change in volume of these lesions of 53 cubic millimeters, which means the average lesion grew a little bit. Whereas in the high dose, we saw that the average lesion volume decreased by about 450 cubic millimeters. So you can see this is a 1% increase in the placebo versus a 10% decrease in the treatment, high dose treatment. Now, that we don't know if that's bad or good. We don't know if there might be another medicine that might decrease this 90%, or if this is about as good as you're going to do with 1 year of treatment. Maybe it takes 5 or 10 years to see a larger reduction. This is very early, and it's important that I give those caveats so that everybody understands there's still a lot of work for us to do. But this is certainly promising that we see the high dose of the medicine having an effect in the direction we would hope, and the placebo didn't really seem to have an effect. I'll also note that our low dose didn't really seem to do anything here either, and that was consistent across really all of our findings. The low dose of the medicine for almost everything we measured looked basically like the placebo, and the high dose looked like it was having, in some cases, some beneficial effects. The next thing we looked at was this thing called the Modified Rankin Scale, or MRS. And this is a tool that's not built fit for purpose for CCM, but it is a tool that's been used across a lot of different neurologic diseases that the FDA has worked with. And bear with me here, these charts are a little bit hard to interpret, but I'll walk you through this. Um, a score on the MRS varies from 0 to 5, where 0 is somebody who has really no change in their function based on the neurologic disease, and a 5 is somebody who has really severe changes in function based on neurologic disease. And there's this standard scale for rating different limitations that people might have. And what you can see here is at the beginning of the study, patients on placebo, we required that these patients have an MRS score of 1 or above. So there had to be something related to their symptoms from the disease. And you see that all the patients had an MRS of 1 or above. Most of the patients were 1 or 2, and then there were a handful of patients that were 3. If we got at the end of the study, the same set of patients who took placebo, what we see is that the number of patients with sort of an MRS of 0 to 1 looks roughly the same, a little bit higher. There's a few less patients that have MRS 2, and now all of a sudden there's a bigger group of patients that have this MRS of 3 to 5. So you see this red group of patients who are getting more symptoms and functional limitation associated with their disease. So now this is not statistically significant, but the trend is that these patients on average got worse over time. If we compare that to the high dose of REC-994, we actually see at the start of the study, before they took any drug, that there were some patients who had severe disease, so this MRS of 4 or 5, a number of patients with 3, and a subset of patients with an MRS of 1. And we see that over time, the patients with the MRS of 4 or 5 seemed to resolve, and you actually had more patients who had this MRS of 0 or 1. So a trend towards improvement. Now, if we wanted to get excited and go to the agency to say, let's get this drug approved in a subsequent study, we would need a lot more patients so that we would have the statistical power to have confidence that what we saw in the MRI and what we saw here was real, and it wasn't just a statistical anomaly. And so if we're able to go forward with a larger study in the future, we'll be bringing more patients in order to make sure that we have powered this study statistically to give us definitive answers. Was this just luck because there's noise and variability, or was this actually a meaningful result? What's interesting is we did some exploratory analyses to ask from all of our data, can we learn anything else? And these weren't things that we pre-specified ahead of time. These were things that we said we're going to go explore after the fact. And one that was really interesting here was to look at brainstem only patients. And the reason we were looking at brainstem patients here, patients who have lesions in their brainstem, is that when we talked to KOLs, they told us that these are some of the patients that are hardest for them to treat. These patients often have lesions in an area of the brain that is just really hard for the surgeons to get to. And this brainstem location, this brainstem is so critical in these core functions for us as humans. And so if you have a lesion and it starts getting leaky, or it starts to bleed, or you get inflammation, oftentimes that translates at a higher rate into severe or problematic symptoms. So these are some of the patients with the highest unmet need. And what we saw here was just like in the earlier waterfall chart, when we look at placebo and then high dose of REC-994, only in brainstem patients from 0 to 12 months, we see that in the placebo, there was a very slight improvement. So basically their lesions were relatively unchanged. They got 0.4% smaller. But in the REC-994 high dose group, we saw that the lesions got smaller to a larger degree. And this is promising because these are patients that are really hard to treat with surgery. When we look at the MRS here as well, we see the same trend that we saw before. We had patients on the high dose of REC-994 that had this MRS of 4 or 5 that seemed to improve. And remember, when these assessments were made, neither the physician nor the patient knew whether they were on drug or placebo. And so it wasn't just that people thought, maybe I'm feeling better because I'm on drug. They really didn't know, and the physicians didn't know at that point either. We also saw that the placebo group, we saw that actually there was this increase in patients with a MRS of 3 and this increase of patients with an MRS of 4 or 5. Now, again, these are not powered statistically for us to say this is definitively what we would see in a larger group of patients, but certainly these are trends that are leading us in the right direction. We did a bunch of other work that we don't have time to go through today. So we looked at something called hemosiderin ring deposition in the MRI. So when these lesions get leaky, blood gets around the lesion in the brain. Blood has a lot of iron in it, and iron shows up on MRIs. And so you can use these tools with MRI to look at how much leakiness has been around the lesion chronically through the amount of iron that exists. This is called hemosiderin. And what we saw was time-dependent reductions in the hemosiderin ring size, like we saw with the lesion size in the high dose versus the placebo. There was some change in seizure frequency, actually a reasonably large reduction in seizure frequency in the 400 milligram arm compared to the placebo. However, I want to caveat this, it's really important. There were some big imbalances in the seizure history between these arms. And so this is very low confidence for us. We wouldn't be able to make any claim here because there was such a big difference in the number of patients and the degree to which those patients had seizures between the placebo and the high dose that it would be really hard for us to take away anything there. We did look at the incidence of new symptomatic hemorrhages across the arms. And what we saw was that there was a very small number of patients with new hemorrhagic events, and there wasn't an obvious difference in these events between groups. But remember, we're talking about very, very low numbers, sort of 1 or 2 patients per group here. So it would be hard without a bigger trial for us to see a difference there. And then we looked at a bunch of other patient-reported outcomes, like PROMIS 29, CCMHI, et cetera. And what we did not see differences in those between the treatments arms of the study and placebo. So lots of places where we didn't see any changes, and those are really important findings as well. So a couple conclusions before we dive into questions. Phase 2 results overall, one of the most important things, Rec 994 was observed to be safe and well tolerated. And this is important because if this is a medicine that ends up one day becoming a tool for patients and clinicians to decide to use together, it is likely to be a medicine that patients would take for a very long time. And we wouldn't want patients to be on a medicine that made them feel awful, especially if it was a medicine that took years to kind of reduce the growth of these lesions or help reduce the symptoms. So it was really good to see the robustness of the safety profile. Now, in a much bigger study, we could see safety signals that we missed in this small trial. So still caveats there. But for the trial we've done now, this was a really, really great result on the safety side. On the lesion size and volume in CCM patients, we saw these encouraging trends in the reduction of lesion sizes with the high dose group. And we also saw these encouraging trends in improvement in function on the modified Rankin score. We've got more work to do there. We've got to do larger studies if we want to prove these things to ourselves. But our next steps are to continue with this long-term extension. So we've got many dozens of patients who continue to stay on the medicine, and these patients are getting MRIs on a regular cadence. And we can ask for patients who stayed on for 2 years, let's say, instead of just 1 year, do their lesions continue to get smaller at a similar rate? Or does that rate increase? Or actually, do we see that it reverts? And maybe this medicine did something potentially in the short term, but not the long term. And we also have to have discussions with the FDA and other health authorities about how they see this data. And if we were going to design a new trial, we'd want to make sure to do that together with those health authorities so that the data we generate from that trial can help, again, not just Recursion, but potentially other companies that come down the road. And then just on a personal note, what I would say is, Rec 994 identification in CCM and the work we did with the Alliance to Cure and in Dean's lab, these early insights were what led us to build Recursion to try and replicate the broad approach, this kind of unbiased screening approach using ML and AI and images and now many other tools we've built. This was the first project we did, and it gave us a sense we might be able to do this for many other diseases. And we now have at Recursion eight clinical trials, eight programs in clinical trials. We've got big partnerships with Roche and Genentech looking at big neurologic diseases and with Sanofi looking at inflammation immunology using an approach that's much more advanced than it was back when we did this program, but is still philosophically very aligned with the work that came out of this. So this one's very near and dear to our hearts. I do want to thank, in addition to our team at Recursion, Janine and Beth and others that have been advancing this work, all of the investigators and clinical sites. So, Dr. Burkhart presented this at ISC, but all of these other physicians were involved, and you can see the various sites here. And then a huge thanks to the Sycamore trial patients and their families. It is a lot of work to be a part of a clinical trial. Each of you are pioneers, and regardless of whether this particular medicine ends up being beneficial to patients through all the work that we have to do in the future, you are blazing a trail for not only this medicine, but medicines that come behind it. And a huge thanks to the Alliance to Cure for their support. It's been absolutely fantastic. So with that, I'll conclude my remarks. I probably went way longer than I was supposed to, but I'll invite